All right, I think we're live. This is excellent. For those of you guys who are joining us here live today, happy Friday or Saturday, depending on which part of the world that you're tuning in from. And if you're going to join us later, that's okay too. I think this live session could serve as sort of an archive. At the least, we have exactly 1 month left to go, or just about. So for those of you guys who will be watching this video later, that's great, because part of what I want to do today is not just to go through the units, which I will, but I also want to connect the things that we're doing in this unit to some of the larger ideas that you need to know for AP Bio. So one of the main things that I'll start off by saying is that in energetics and of course cell communication, cell cycle, all of these different topics can get pretty detailed, especially when you look at things like the Calvin cycle or even the Krebs cycle. There are so many substrates, so many enzymes, and many students get very, very confused as to, well, what is the depth at which I need to understand this stuff? So that's sort of my purpose here today. I will review those ideas in general, but I'm going to be focusing mostly on exactly what you might need to know for the exam, the depth at which you need to explore these ideas. Okay, so it's going to be pretty challenging because we're going to be doing this for a couple of hours today, trying to get through, about 5 chapters. So that's definitely faster than I've ever done it in my life. So I think this will be a challenge, but also kind of interesting at the same time. I'm just getting a little warning here on the studio. I'm not sure what's going on. Maybe there's a bit of a delay or something. I'm not sure. So hopefully we're not having too many technical issues today. The internet has been a little bit wonky. All right, enough of that. Let's get started. All right, so the first unit that we're doing today is Unit 3, which is on energetics. So this unit actually encompasses 3 chapters of Campbell biology. So if you guys are studying from Campbell, then these would be chapters 8, 9, and 10, at least in the editions that I worked with in the past. I'm not sure if those have changed, but the primary idea is that chapter 8 is supposed to be about energetics in general. So what is, let's say, biological energy? And also it contains ideas about enzymes, which is related to energy, as we'll see today. Chapter 9 and 10 deal with cell respiration and photosynthesis in that particular order in the textbook. But in most of the classes, as per the AP Biology CED, it actually goes from photosynthesis and then it goes over to cell respiration. So we're going to also do it that way because for me, I think that makes a lot more sense. We want to know what the source of that initial energy input is through photosynthesis and then break it down in the cell respiratory process. So this is that chapter 8 that we're going to begin with. It's got a ton of stuff that you don't actually need to know. So what I'm going to do is try to kind of focus on the main ideas, and it sort of jumps all over the place. This actual chapter is a little bit jumpy. But the first thing that I want to talk about, I'm going to skip over a lot of slides here too. You can't see what I'm doing, but I'm going to skip over to this slide over here. Because the way that we should be linking what we're doing in chapter 8 with what we're doing in the previous unit, which, by the way, if you remember, ended with cell membranes, is that we learned about diffusion. We learned that if you have substances that are all highly concentrated in one part of the system, that it has this tendency to diffuse out. And the idea that we applied with this diffusion was the transport of materials across membranes. And hopefully you guys will remember that when you have this transport of materials from an area of high concentration like we have up here to an area of lower concentration like we have down here, then that was free. Or at the least, it didn't require any energy. And in that chapter, we also learned that when you take all of these materials that are separated and we try to move them towards an area of higher concentration. Now, that is to say that we're doing active transport that required ATP, or energy. So why is that happening? And the best way that I can explain it is actually just looking at this picture on the left. You can ask yourself, why is it that gravity pulls people towards another massive object? I mean, we could get very esoteric with physics about that, but we don't need to. One of the things that I'll say for AP Bio is that all we need to know is that going from an area of high concentration to an area of lower concentration is just one of those things that the universe prefers to do. It's just like a pencil falling from, the top of your desk to the bottom. It just happens. And therefore, it's going to be not only free, but it has this tendency to release energy. Because when we look at this diver as he dives into this water, we know that there is going to be release of energy in terms of kinetic energy that's being manifested, as well as the splashing and just the disruption to the water molecules. We know that energy is being released. Now, why any of this matters is that largely this diffusive property that we see in the middle can be thought of as something that we call entropic change. Because we know that entropy, at least in terms of physics, generally means the randomness of the system. And the best way to think about it is that when you have all of these molecules that are clustered together on one side, that's not very random. There is this degree of order with which these molecules are being held together. And as entropy or the randomness increases in our universe, well, that's the type of, let's say, direction that the universe prefers to go. All right, now I know that that's a lot of what we've been doing in the last unit, so let me connect this to what we're going to be doing today. You can see on the right side that instead of looking at molecules that are all just sort of concentrated in one part of the system, we can think about atoms that are actually, well, concentrated, for lack of better words, into a single molecule. Now, if you are doing chemistry concurrently, or if you've done chemistry before, then what we know about this molecule up here is that there are a bunch of atoms together because of the fact that there are atoms that are bonded together. And in the case of this particular molecule, which appears to be glucose, C6H12O6, these are all nonmetals. We know that they're going to be bonded via covalent bonds, or the sharing of electrons. We can make a similar claim that this glucose molecule has relatively low entropy relative, again, to what we would get if all of these molecules were separated as water molecules or carbon dioxides. The water over here with the reds and the light gray, and then this right here, that linear molecule, that's the CO2. They're not actually keeping the conservation of mass here, but you can imagine that if all of these atoms were to be separated into simpler molecules, that would be favored by the universe. And that's what this picture is pointing at, the favorability of breaking things down. Now, we relate a lot of different ideas into this picture, and those ideas are all connected together, and I think it might be helpful to know. Now, you don't need to memorize any of these terms for AP Bio, but the generalized ideas, and I'm going to link this to other units later, but the generalized idea is that when you go from complex to simple, this should release energy, as we saw with the diver. So we call that an exergonic reaction, because X as in out, there's energy coming out of that molecule into the environment. So again, we know that when glucose gets broken down, there's energy being released. All right, so that's fine. The fact that we're going from complex to simple means that it's a catabolic reaction. And the way that I like to remember that is I imagine a cat, being released into a room where there are all these Christmas trees and it's just breaking things down because cats can be crazy sometimes. So a catabolic reaction breaks things down, and it's going to be exergonic, and it's also favored by the universe, and that's how we got to think about the breakdown of glucose. Now, we can also, again, go back the other way and say, well, if we're to take these CO2 molecules and these H2Os and we want to combine them together into forming a glucose molecule, well, that's going to require energy. So what is that? Well, that's an endergonic reaction, because in this case, enter, there's energy entering the molecule, and it's also going to be an anabolic reaction, just because if it's not catabolic, it's got to be anabolic. There's really no good way of memorizing that. But the most important thing is that there is this energy that needs to go in. And what we learn in this unit is that this picture is sort of a preview for what we're going to be doing. Because photosynthesis is that very reaction, or the series of reactions anyway, that take these simpler carbon dioxides and water molecules and then get them into that glucose form while getting that energy from sunlight. And that is why this picture is so important. Now, I did say that I wanted to relate this to some of the larger ideas in biology, or at least in AP Biology. So I'm going to jump ahead here to something that I prepared for you guys. I looked through some of the FRQ from the previous, maybe 5 years or so, and then I picked out some questions that may relate to what we're dealing with here today. Actually, there's a good chance that I didn't get this question in particular. Oh, that's too bad. Okay, I'll find out later. Maybe I put it right after photosynthesis. All right, no. Okay, sorry about that, guys. So I thought I was thinking about a question in my head. I'll just explain it to you guys, what the question was. So the question was very general. I think it was from a couple of years back, and it says something like, what is, maybe it was 10 years ago. I'm sorry, it doesn't matter. The question was, what is a process through which plants can gain mass If they're in sunlight, what is the process through which plants can gain mass? And what is an inorganic substance that can contribute to that increase in mass? So it's a very, very broad question, right? They're not asking specifically about, what cytochrome are doing or what photosystem one's doing. They're just saying, how does the plant actually gain mass? And for us, it's just looking at the reversal reaction of this change right here. The fact that these carbon dioxide molecules or water, see, they could have, you could have answered either one of those, is going to eventually contribute to the formation of glucose. Now, for those of you guys who have done a lot of review, you know that water is actually only contributing electrons that form the bonds between the glucose molecules, but, electrons are mass, so that still counts to a certain degree. And then they also talk about the reversal reaction. So if the plants are losing weight in dark, why are they losing weight or mass? And that's because they're also respiring and they're breaking things down, and then the gas, CO2, is being released back into the atmosphere. So anyway, I wanted to talk a little bit about the changes. So again, I said that I would relate this to some of the larger ideas in other parts of biology, so I'll do that right now. One of the main things that we think about when we look at things like macromolecules is the idea of polymerization. So if you guys remember talking about how polymers form, they form as a result of monomers that start to combine together. So you have basically maybe amino acids combining with another amino acid, which then combines with another amino acid. And essentially what you're doing in that particular situation is creating complexity, because obviously having just a few amino acids is going to be simpler than having a polypeptide of hundreds of amino acids. So that in itself is an endergonic reaction. So when we link that all the way to Unit 6 and say something like, well, when we do translation during the gene expression pathway, ribosomes are actually combining all of these different molecules together in order to form that larger polypeptide. Well, that actually directly links to this picture as well. Because what we're doing in that situation, once again, is doing an energetic reaction that requires, well, in that case, ribosomes. And in the case of translation, it's requiring the form of energy that we call GTP, but that's also pretty close to ATP anyway. And speaking of ATP, let's have a review of that too, because this is also one of those things that are incredibly important for AP Bio. Now, I think it goes without saying that ATP is commonly known as the currency of energy, and that's fine. Currency of energy is just sort of the dollar that we can spend at any of the stores that we might call chemical reactions. Now, what's important about this is that, again, the use of this ATP, as shown in this picture, directly links to the idea of entropy and the endergonic, exergonic, all those ideas that we've been just talking about. Because when you look at the structure of adenosine triphosphate, we have an adenine, we have a sugar, so this would be the ribose sugar, and we have 3 phosphates. And for the purpose of AP Bio, once again, we can think about this pretty simply and say, all right, well, when ATP breaks down, again, I'm using that term breaking down because it's a catabolic reaction, and it breaks down into a single phosphate that's inorganic because it's no longer attached to an organic substrate. And you have ADP, so adenosine diphosphate. Well, that is in itself an exergonic reaction. And it has to be, because it is going to be a catabolic reaction that simplifies things. This is favored by the universe, and you see this energy coming out. Now, there is this fixed amount of energy that is calculable in terms of joules, but you don't need to know that for AP Bio because we're not doing biochemistry in greater detail. So that's fine. We have energy coming out. So the idea of ATP being broken down into ADP, once again, is that catabolic reaction. And if we were to charge this back up to ATP, because, that's what we do, right? We have cell phones that we use, the battery goes down, we charge it back up, the cells are going to do the same thing. They're not going to be wasteful. Cells are very, very, very frugal with the substrates that they have. So we're going to be able to recharge that, and that reverse reaction, of course, would be, in fact, that cell respiration process, at least the phosphorylation processes that we see throughout the Krebs cycle, as well as glycolysis, and of course, oxidative phosphorylation. So I'm actually using all those terms because I do feel like if anyone's watching this at this point in time or in the future, clearly this is being done as a review. So hopefully none of the things I'm saying is new to you, because if it is, then I feel like perhaps looking over this unit before watching this review video might be more beneficial. All right, great. So that's a little bit about energy. Now, we can link enzymes to this picture, so let's go ahead and do that. So I like this picture because this is, again, something that AP Bio likes to use as example, sucrose or any disaccharide, right? Because we also talk about lactose. That's in the lac operon from our gene expression regulation. But sucrose is one part glucose and is one part fructose. So this is a relatively complicated molecule compared to what we see down here with glucose and fructose as separate. And the idea is, again, you have a hydrolysis reaction that could break this sucrose down into their simpler forms. This would technically be a favored type of reaction. However, the issue is that what we see in the real world is that you can put a bunch of sucrose into water, and you can do this at your house because sucrose is just white sugar. You can put it into a glass of water, and it will not spontaneously break down into glucose or fructose. So even though it's favored, it's not going to do it. And it's sort of like a ball sitting on top of a hill, right? It's sitting on top of a hill, and it's, at a higher gravitational potential energy than it would be if it were at the bottom of the hill. But it's still sitting there because there's just nothing really pushing it over the edge. And the idea is for enzymatic reactions, even favorable enzymatic reactions, where we start with maybe sucrose over here and glucose and fructose down here. We know that this is a catabolic reaction because the energy is coming out of that molecule, so we have an energy decrease in the molecules. But this hump right here that we call the activation energy is what hinders or inhibits any sort of reaction from proceeding forward. And in the case of catabolic reactions, you still have this the activation energy that will inhibit this reaction from going forward. So the main idea then is by having sucrase, and as you guys may want to know, anything that ends in ASC would be an enzyme and it would be a protein. This sucrase has the capacity to bring down that activation energy. It's almost like this little nudge on that soccer ball at the top of the hill so that it starts to roll down that hill and release that, once existing potential energy. So the decline in that activation energy is what enzymes are primarily responsible for in biology. So here's the thing. The problem with thinking about the reduction in activation energy is, once again, that we're dealing with energy. This is something that you could theoretically calculate. But again, for the purpose of AP Bio, what they want us to know is, in general, not about math, but in general, how does that activation energy actually decline in a macroscopic manner? So here we're introduced to something called the induced fit model, which deals with how enzymes work. And I can't stress this enough because I'm going to be talking about this picture for quite some time. It's very, very important because the way that enzymes operate is something that AP Bio will ask almost every single year, whether it's on FRQ or on MCQ, it's always going to be there. And let's actually look at the phenomena first, and then we'll talk about why this matters so much for AP Bio. So firstly, we have an enzyme, and by this time, I think you guys would know that this is made from a protein. So this may be a single polypeptide or it could be multiple polypeptides, but ultimately it's folded into a shape that lends itself to having something called an active site. And the main idea is that the substrate with which this enzyme is going to interact, so that would be maybe sucrose if this were to be sucrase, it has this affinity for the active site. And whenever I say affinity or any sort of, these sort of words that tend to kind of seem anthrocentric or whatever, what I'm trying to say is that there is a chemical affinity, right? There's this chemical interaction that is likely to happen as that substrate engages with the active site. Now, the induced fit model states that this active site and the substrate is actually not a perfect lock-and key type of fit. Even though we talk about it that way in 9th grade bio, in the induced fit model, we say that they actually are not perfect, but as the substrate binds to the enzyme also, in return, binds to the substrate. And you can sort of see this a little bit more clearly in this picture where the active site is sort of open, but as the substrate binds, the active site sort of closes in on itself. Now, I'm going to talk about a little bit more detailed analysis of what's happening. Not that you need to know all of that for FRQ, but I think it forms a really good foundation for how you're going to justify your answers or rationalize answers that deal with this particular question. But the question always is, what is it about enzymes that allow it to operate and do what it's doing? So they might put this into a specific context. They might say, well, what is it about the structure, or what is it about luciferase or something that allows it to convert luciferin to oxyluciferin, right? Whether you know what those substrates and the enzymes are, it doesn't matter. And I think that's part of where AP biology students have so much trouble because they're like, we never learned about luciferin. Well, the point is not about luciferin. The point is that in the grand picture of things, in all of biology, enzymes are going to behave in the same way according to the same ends that it's trying to perform. So let's actually have that conversation. The first thing that I want to say is, how is it that this induced fit model is working? How is it that the substrate can impart or induce this change in the enzyme as well? So we go all the way back to Unit 1, because in Unit 1, we learned that in macromolecules, proteins, as we know form enzymes, are going to be comprised of a lot of amino acids. And each of these 20 amino acids have a very special chemical group that we call the R group. Now, they can be largely categorized into things like polar R groups and nonpolar, and then we also have acids and bases. But you may recall that when you combine all of these amino acids in a linear chain, there's these, side-by-side interactions of these R groups, or perhaps even at distance interactions of these R groups in such a way that the polypeptides begins to fold. And that is the technical term. We call it protein folding. So the polypeptide begins to fold, and it's really folding in sort of the rules that are dictated by physics, right? Because these enzymes are primarily being created in the aqueous environment of the cytosol, so there's water around it, so the hydrophobic guys are going to want to go in, and there's this idea of conforming to that environment. In fact, when we talk about protein structures, we call it conformation. We call it a protein's conformation. But at the end of the day, what's important to note is that the conformation of a protein is driven by the sequence of those amino acids, and it's driven by the chemical interactions of those amino acid R groups, and even some of the side chains that can form secondary structures. So if we can establish that a polypeptide structure is primarily driven by the chemical interactions of substances that are forming this enzyme, then by the addition of another substrate, which presumably is also a chemical, you're going to now have additional chemical interactions that are being placed upon this polypeptide at the active site. That is to say that if you have this substrate that binds to the active site, it is now an additional piece of chemical that can have an impact on the overall structure of the protein. So when a substrate binds to an active site, we actually have a very specific term that we use. We say that this imparts a conformational change on the protein, or a conformational change on the enzyme. It doesn't really matter. In this context, we're talking about the enzyme. And why that's important is that now the change in the structure of the protein can do all the things that reduce the activation energy, which I'll mention in just a few moments. But this idea is so important because it's all about structure and function, right? When the structure of the protein changes, it becomes now functional in doing what it's supposed to do. Now, how does this relate to some of the other ideas? Well, thankfully, I actually have another slide up here that's going to help us engage with that idea. So but I guess ahead into G protein coupled receptors, which is really part of the next unit. This is going to be mostly about chapter 11, I think, cell communications. We see that a G protein coupled receptor can bind to a signaling molecule. And this is really not that much different from a substrate binding to an enzyme, right? It binds to the G protein coupled receptor. And what that does is that, again, it causes the chemical interactions to change the structure or the conformation of this G protein coupled receptor on the inside. And it's that change that can potentially knock out that GDP from that G protein, allowing that second, or at least the activation of that G protein in order to, let's say, go ahead and activate adenyl cyclase or whatever it is that it's supposed to do. This is, of course, a very general picture. So what we're seeing is that receptors, much like proteins, are going to be very, very sensitive to other molecules binding to it, and that conformational change that changes the structure of the protein, that is where AP Bio is going to primarily ask questions. Okay. All right. Now, I did say that I was going to talk about how the active site attachment of the substrate can reduce activation energy. That's one way to put it. What happens is that you could have substrates that are oriented correctly. So, I mean, this sort of seems silly, but let's say that you have a particular direction in which the two substrates are supposed to bind. But, if they're just randomly bombarding into each other in a system, then the chances that they're going to bombard into each other in that particular orientation is very low. However, if the enzyme itself has an active site in such a way that it can bind one substrate and then bind the other substrate, but they have to be oriented in a particular way, then you already have your work cut out for you because they're in the right orientation then. And that in itself actually can reduce the activation energy. Now, that is, again, calculable, but you don't need to know the calculus of what is actually being reduced. It can also bend bonds. And again, bonds are physical, so you can strain substrate bonds. You could even provide a favorable microenvironment. So what does this mean? This means that enzymes, like, for example, in the picture here, well, maybe there's a bunch of acidic R groups that are associated with the amino acids that form the active site. So what does that mean? Well, that means now in this little microenvironment, you have greater acidity, and who knows, maybe that actually works out for the substrate reaction. So that's going to help out as well. And then sometimes we even have temporary covalent bonding to the substrate. But ultimately, all of these are going to really help out with reducing that activation energy. So what else do we need to know about enzymes? Well, after the enzyme has done its job, you should probably know that enzymes can be reused over and over and over again because they're not actually used up in the reaction. Now, everything in biology does have a lifespan, which means that the proteins will eventually degrade due to, well, actually, it's fairly well controlled by the cell, but as long as the enzyme has not been broken down by external means, the reaction in itself is not going to do that. So you can use it over and over again. That's great. Now, this is something that we have talked about in Unit 1, because in Unit 1, we have this idea of denaturation. Again, denaturation of enzymes is something that always comes out on this exam. They actually tell you that there are 3 environmental factors that can cause denaturation, but really 2 of them are tested on this exam. So that's either going to be temperature or it's going to be pH. Again, the idea is the third one, which is salinity, is pretty important, but it's not something that we see a lot. I think I do have a question on this, so I'm going to search for that as I explain what's going on here. So when we talk about either temperature or pH, the main focus of these questions are going to say something like, what's going on here? Why is it that enzymes are doing poorly outside of their optimal range? And again, the optimal range is sort of this peak of this bell-shaped curve there, rate of reaction on the y-axis. The main thing that you want to say is that outside of their optimal range, the structure begins to become compromised. And without structure, they don't have function. So once again, that structure and function is going to play an important role even when we talk about enzymes. So then you can go into a little bit more detail, because if you just write that on your FRQ, I think you're cutting yourself a little short, and also the readers might not give you the full points, because the next part that you have to talk about is another generalized biological fact that, well, why is it that the enzyme structure becomes compromised? So you say something like, well, if it gets too hot, let's say, and that's typically the way that we think about it, if it gets too warm, then what ends up happening is that all the chemical bonds start to become sort of weakened. And we know this because when we talk about things like denaturation of DNA, we know that when you turn up the temperature in which DNA double strand is existing, it will start to kind of fall apart, right? It will fall apart into 2 single strands. And this is sort of the basis for how we run PCR for those of you guys who have studied that, right? You have a thermocycler that increases the temperature to like 90 degrees, and it actually denatures DNA. Now, if you want to get really, not really, but if you want to get a little bit more detailed about it, the idea is that all of these bonds are being formed by the interactions between atoms. Those atoms all have a vibrational energy that's associated with the kinetic energy of the system. But as that kinetic energy increases, that's what temperature is, then the vibrational energy becomes so high that things like, hydrophobic interactions, I'm sorry, I take that back. Oh my God, hydrogen bonding. I can't think this morning. Things like hydrogen bonding, which are weak intermolecular forces, they're not going to exist anymore. And then if you turn the heat up a little more, then ionic bonds start to break down. And you turn it up even more, then even covalent bonds will break down. So the idea is outside of that optimal range, or at least when you turn up the heat, the structure starts to fall apart because the bonds start becoming broken. And to be honest, on the AP bio, you can just say the bonds become broken or become compromised is going to be sufficient. Now, for whatever reason, if they say, well, why is it also not as efficient at lower temperatures, then, well, there we're talking about the reduction in kinetic energy, and there's just less movement of any materials in a system where there's low heat, so you might not actually see that great degree of bombardment or at least engagement of the substrate to the protein. Now, in this picture, we all, of course, see that enzymes could adapt to higher temperatures, and in this case, thermophilic bacteria. So these are, these are organisms that may live in maybe hydrothermal vents or those very, very hot springs in Yellowstone National Park. While these guys, they've evolved, right, over the last however many billion years to have proteins that are specifically catered to resisting denaturation. So that's what we have here. And by the way, again, I refer back to PCR, but the reason that PCR can do its job is, of course, we're using a DNA polymerase from Thermus aquaticus, which would be a thermophilic bacteria as well. pH is a pretty similar story. I know that for chemistry people out there, you know exactly what pH is, but for those of you guys who are not so keen on chemistry, like me, I was never a big fan of chemistry. The only thing that you have to think about with pH, actually, there are two things that you need to think about with pH, is that, first of all, things get basic as you go beyond pH of 7, right? And things get more acidic as you go below pH of 7. Like, you have to know at least that for the AP exam. And the second thing is that when something is acidic, it means that there's a lot of H plus ions. Now, I know that technically it's about the relative ratio of H plus to OH minus, but you can, for the purpose of AP Bio, just think about it as being, well, there's a lot of H plus ions in acidic solutions. And in basic solutions, there's not a lot of H plus ions. Or perhaps there's, a lot more OH minus ions. But the point is that whether we're talking about H plus or OH minus, the charges that are associated with these different ions is, of course, going to have impact on the chemical relationships that we see between R groups. Because we said earlier, there's hydrogen bonding, and then there's also ionic bonding. Well, what are hydrogen bonding and ionic bonding? Well, these are actual electromagnetic interactions. Hydrogen bonding is the interaction between slightly negative and slightly positive poles of molecules. Ionic bonding is between something that has an extra electron versus maybe something that's missing an electron. So we have these charges. Once again, we're looking at electromagnetic interactions. So when you enter into this, a lot of protons or H plus ions and a lot of OH minus ions outside of what we normally would see in the context of how this enzyme would operate, clearly you're going to have some compromisation of those structures, and that is going to explain why these pHs are going to impact it too. So let's see. Do I have a question on this? I don't think I have a question. It's so weird. I thought I had collected all these questions that deal with this, but I think they were just so old that I didn't really do that. Okay, so let's go ahead and tackle the more important stuff. Okay, so we have about, I would say, half an hour left to talk about photosynthesis and cell respiration, but I think that's fine because, setting all of this stuff up is more important than anything else. And by the way, things like, how negative feedback works and how positive feedback works, very, very important for this test. Once again, if I were to just point out all the important bits, this is something that you absolutely have to know. Being able to detect that negative feedback takes the final substrate and, diminishes the pathway, whereas positive feedback will accelerate the pathway. Those ideas are, of course, very important. All right, so let's talk about photosynthesis. So the good thing is we did spend about half an hour building the groundwork in explaining that what we have in photosynthesis, of course, is going to be an endergonic reaction. And it's going to be a reaction or series of reactions that combine inorganic carbon dioxide and water into a more complex molecule such as glucose. And we also have the byproduct, oxygen. Now, you'll notice that in our typical photosynthetic reaction, we have 6 CO2, 6 H2O, yielding 1 glucose and 6 O2. And technically speaking, there are 12 and 6. Again, you don't need to know that. It's just that 6 of those water molecules are canceled out in the product side. So you just have 6 CO2 plus 6 H2. And that is completely sufficient for the detail that you need to know for photosynthesis. All right, so what's the main idea? Well, so the first thing that we need to do, I think, is to understand what it is about energy that we can extract from light, right? Because earlier we said that this photosynthetic reaction is going to be an anabolic reaction. It's going to require energy. So that energy, which comes from the sun, is going to be in this visible light spectrum, right? So we have 380 nanometers all the way to 750. These are the colors of the rainbow, as you may remember from kindergarten. You have Roy G. Biv, so you can go that way as well. But what's important to note is that what this shows is this idea of energy. Now, it's missing this frequency thing, but that's okay. The shorter the wavelengths, we say there's a higher frequency of that photon, which then allows it to have higher energy, and it will be the opposite in the longer wavelength side of things. Now, the problem with thinking about this is that we need to understand that energy is not a biological thing. It's not a chemical, right? Energy is not something that you could weigh or figure out the volume of. It exists in a slightly different domain to the matter that we care about in biology. So the first thing that I think we need to do is to talk about how energy actually engages with matter, where the interface of that light energy is. And that is where we introduce the idea of pigments, right? Because whenever we talk about pigments, and this could be biological or not, pigments are substances that have the capacity to selectively absorb certain wavelength of light. So, for example, if you have a red T-shirt, the reason that it's red is that the pigmentation that has been used to dye that red sweatshirt or red T-shirt absorbs pretty much all the wavelengths of visible light except the red ends of that spectrum. So that part is reflected back or sometimes transmitted, which is what's detected by our eyes. So the pigments that we're going to be seeing in plants, which generally you can think of as chlorophyll, right? Because they're not going to really differentiate chlorophyll A or B on the exam. So when we say plant pigments or chlorophyll, we know that chlorophyll pigments appear to be green. And it's sort of like a greenish yellow, really, right? So we have a green pigment, which means that this is a pigment that can absorb the blues and the reds of the spectrum, reflecting back what we have right here in the middle. Okay. Now, what does it mean to absorb that energy, though? Well, the absorption of energy operates something like this. So we have a magnesium atom. And by the way, I'm going to try to simplify this for a better understanding. So there's a lot more technicality involved in this. But when we talk about magnesium, this is an atom. It has 12 protons, and it's got a bunch of neutrons. Has all these electrons around it. If you think about a Bohr diagram of a magnesium, you have the nucleus and all of these electrons surrounding it. What's really happening is that when light hits that chlorophyll molecule, the energy of the light has the capacity to interact with that matter for the first time. And the interaction of light with that matter, it presents itself as an electron becoming excited. Now, this, again, is a technical term, even though it sounds kind of weird. But an excited electron is an electron that's been sort of stripped away from its initial ground state. So, for example, if you have an electron in the first shell or the second shell, that's where it wants to be. But you take this electron and it absorbs this light energy. It jumps to a higher energy level. It jumps to a higher shell. Kind of like if you were to throw a baseball up in the air. It jumps to a place where it doesn't want to be. And why this is so significant is that, as you know, with the baseball, if that baseball fell right back and hit your head, that's going to hurt because it releases that energy back in a different way. And the idea is that when light makes this electron jump to a higher energy level, it could come back. But the problem is that what we're seeing in this picture, right here, right now, the one that I'm showing you, is actually not what the plants want to do. I'm going to explain that in a minute. But look at this electron. It gets excited, and then it falls right back down. And as it falls back down, it releases heat and another photon of a slightly different wavelength, which is a function of the lost energy and the frequency, and then it just releases a different color, and it happens to be violet. Now, this is what we call fluorescence. But as I mentioned, this is not what plants want to do. In fact, we only see this type of fluorescence in cases where we extract a bunch of chlorophyll molecules, we put them into water, and then we are shining light on it, and then we see it fluorescing. Now, why is this something that plants don't want to do? Well, the plants don't want to do this because when you just simply have that excited electron absorb that energy of light and then have it drop and release this heat and photon, you've done nothing with that energy, right? The purpose of photosynthesis is to actually use that energy to make sugar, but you've just kind of released them as heat and another photon. Nothing has been done. It's, again, like that baseball hitting your head. You've accomplished literally nothing. So the reason that this picture is being shown is to demonstrate that what we actually have in the real world is not just chlorophyll molecules that are randomly dispersed throughout the cytoplasm of a plant cell, is that they are actually carefully connected in an array of chlorophyll molecules, such that that excited electron does not drop right back down, releasing heat and releasing another photon, so that we don't waste that energy. Because we want to harness that energy, or at least when I say we, I mean plants, and ultimately we too, but we want to capture that energy. So the way that I like to think about it, and again, I'll introduce this very complex protein called photosystem later, but what's actually happening is that you have almost like a volleyball team, right? Because if any of you guys play volleyball for school or for fun, what you know is that in volleyball, the point of the game is to not let that ball drop on your side of the court. So as the photon comes and as the electron gets excited, that electron is sort of like the volleyball that's going up in the air. And you want to volley it, right? And that's why I call it volley. We volley it, volley, and then you get the setter. And then the setter puts it up. And after that point, it gets spiked. Okay, now we're not going to get to that point just right now, but just know that that's what this is all about. The antennae chlorophyll and the special pair, which are the setters, what they're doing is they're capturing that excitation energy from the electrons, the energy from the light and then excited electrons, and they're trying to preserve it to the best of their ability so that it doesn't get wasted. All right, great. So that's sort of the interface of energy and matter that I wanted to go with. But now we can look at sort of the big picture stuff. So let's go ahead and look at the macroscopic image of where this is all going to happen. So as you guys know, this is a chloroplast. By the way, a few things in this picture that's very important. Chloroplasts have a double membrane. And what does that mean? Well, that means that endosymbiosis theory, which stipulates that chloroplast was at once once upon a time a free-living prokaryotic cell. Well, that's one of the pieces of evidence that supports that. Another piece of evidence, by the way, is that chloroplasts have their own DNA. Now, I want to just kind of, and this is a side note, I'm going to go on a lot of tangents, so hopefully, you'll be able to keep track of what I'm thinking. But the other piece of evidence, when we talk about mitochondria and chloroplasts and the fact that they are non-endomembrane system organelles, and they are endosymbiotic organelles, is that they also have their own DNA. So we know that mitochondrial DNA is a thing. How do we know that? Because in Unit 5, when you do pedigrees, you often run into a pedigree that is called the mitochondrial inheritance pattern. And in mitochondrial inheritance patterns, what you get is this sort of a maternal lineage where the moms give it to, or give whatever disorder it is or whatever condition it is, to all of her children, both sons and daughters. But the sons never pass it on, but her daughters do, and their daughters do. So you see all of this maternal inheritance pattern arising in mitochondrial inheritance, and we learned that one of the reasons that's the case is the mom contributes the egg. And the egg is the more expensive of the gametes, right? I mean, eggs are, of course, expensive. We know that's kind of on the news, but eggs are the more expensive of the gametes. Sperm is cheap. The sperm only contains really the nucleus with the haploid DNA. The egg contains not only the nucleus, but also all of the organelles that's going to contribute to that very first cell that we call a zygote. So clearly the egg is bringing the mitochondria. Now, don't get me wrong, the sperm cells do have mitochondria, but that never enters the egg. And that's the difference. All right, so why does that relate to this? Well, because I've seen in the past, AP Biology pull something kind of shady, all right? Now, if College Board is ever going to watch this, they're probably going to sue me. But anyway, no, I'm just kidding. It's not libel. It's a little bit shady, but I'll tell you what they're going to do. So they say, look, we know that in the AP curriculum that you guys don't do a lot of plant physiology. You guys don't learn about plants to a great degree. It's mostly animal-centric. But I'm going to say that I'm going to ask about pollens and ovules. And I'm going to say, look, if a pollen of an infected plant is used to fertilize an ovule of a non-infected plant, none of the offspring plants have the disorder.
However, if you use an ovule and fertilize it with ovule of an infected plant, but fertilize it with the pollen of an uninfected plant, then you see that all the offspring have the disorder. So what they're really getting at here is a chloroplast DNA disorder potentially, or even a mitochondrial DNA disorder. But instead of using, let's say words like moms and dads or mothers and fathers, they're actually using terms like pollen and ovules because we're not familiar with that, right? Because I mean, we know pollen. I mean, it's allergy season right now, so it's driving me crazy. But we know that pollen is sort of like, we know that it's associated with like the male, and we know that words like ovule, female, but it doesn't connect directly. And College Board knows that, right? They know that students are going to be kind of thrown off by that. But again, just be on the lookout for that. All right, coming back to photosynthesis. Holy crap, that was a big digression. But coming back to this photosynthesis, we know that there's stroma, which is sort of this jelly-like matrix. And then we know that we have a thylakoid or a set of thylakoid membranes that are stacked up into a grana. Or into grana, or granum for singular, it's Latin. And that photosystem that I was talking about earlier, that large protein, which I'll show you again, that large protein is actually on a membrane, right? It's on a phospholipid bilayer. And this phospholipid bilayer, as you can see here, is the thylakoid membrane. So that is where things are taking place. Okay, good. So let's actually take a look at this in the big picture, and then we'll get photosynthesis done, or at least the light reaction done very shortly. So we're going to be starting with the light reaction. As you guys know, we have light reaction and the Calvin cycle. Light reaction in this case, again, we have these two photosystems, photosystem 2 and photosystem 1. The numbers are backwards, but I'm sure if you have a teacher who likes to tell stories, they'll tell you that they discovered this one first, and then they discovered that one, and then they discovered that the order in which they occur is 2 first, and then 1. They didn't want to change the name. That's fine. But what's important is this portion where we say, look, here's light. All right, the light is going to excite an electron, and that electron gets volleyed. It gets set. We got to that point. And I said, look, right here, this is going to be the spike. And there we go. Along this electron transport chain, the electron gets spiked. But here's the thing. When you, again, I don't know if any of you guys are volleyball players out there. If you spike a volleyball, that's going to go with some force, right? That ball is moving. It's going, right? And if you have a really good volleyball player, that thing is moving very fast, carrying a lot of energy. This electron that gets spiked is high in energy, okay? It's high in energy, and what you see here is that through these electron transport chain proteins, that energy is actually slowly drawn out, okay? So unlike a spiked ball that goes really fast, here we're actually going to say, all right, hold on, let's slow it down. Let's get some of that energy. And what that energy is doing, well, remember, energy is going to be associated with things that the universe prefers not to do. And what we're doing here is concentrating or pumping these H plus ions or protons from the stroma and packing them inside the thylakoid space. And when I say packing them, we're increasing that proton concentration to a great degree in this thylakoid space. And that requires energy because remember, there's low concentration of protons out here. There's high concentration here. If we're moving them from low to high, we talked about it, that's active transport. But remember, active transport required ATP. But here we're not using ATP. We're using the energy of the electrons because they were in themselves energized by sunlight. Great. So we get this electron to plasticyanine, so PC here. Presumably, because they've pumped protons, now those electrons are at a lower energy state. Right? It's just sort of how energy operates. And it gets fed into photosystem 1. And in photosystem 1, at one point, it will have an opportunity to be re-energized by light. And what that does is that it gets passed on to FD, which is ferredoxin, and it goes to NADP plus reductase. But I'm going to pause there because there's something that I didn't talk about. I should do this. I should have actually paused here. The idea of the high concentration of the thylakoid space with these protons means that they want to leave. Okay, they want to leave. So there's a couple of things that we need to consider. Firstly, it's sort of like blowing a balloon, right? When you blow a balloon, you get all that air into that balloon. It wants to leave. It's sort of the same idea because you have all of this compacted air at higher pressure. It wants to go out. Here, this is not quite the same type of pressure, but it's a chemoosmotic pressure. They want to go out. But what you have to understand for AP Bio, and this is so incredibly important, is understanding why this proton cannot go out through the phospholipid bilayer. Why it can't do it. Because again, there's going to be so many questions that pertain to this or at least to play on this idea. So, again, we have to go back to the basics. All of these little orange or yellow tails, what are those? Those are fatty acids. Those are lipids. They don't like things that are charged nor polar. And a proton or an H plus ion is by definition charged. So it can't go through the lipid bilayer because it will not be able to go through the lipid area. So kind of like how when I gave that balloon example, that air can't pass through the rubber because the rubber is, the structure is too small for the gases to escape. It's the same idea. We're able to use or leverage the phospholipid bilayer to build a concentration gradient that we can control. And what do I mean by that? Well, we can control the exit of that proton. Kind of like if you have that balloon again, I'm going to use that example. You can sort of control the rate at which the air comes out, right? You can kind of like undo the balloon a little bit, the lip of the balloon, it goes, or you can be like, and it all comes out. You can do that. And that control is being done by a very important enzyme called ATP synthase. Because this ATP synthase is going to be a channel through which these protons can come out. But because these protons are coming out, much like the air coming out of the balloon, there's a release of energy. The potential energy that is held by the concentration gradient gets released. And remember, energy can be utilized to do endergonic reactions. And the endergonic/anabolic reaction that we're doing is combining adenosine diphosphate and an inorganic phosphate into ATP. That's what we're doing. So it's almost like, hey, we're going to leverage active transport to make ATP instead of using ATP to just do active transport, right? It's all related to the last unit. Very important. And also to the unit before with why the phospholipid bilayer can be used in that type of context. Great. So now that we have ATP, that's going to go to the Calvin cycle, but I kind of pause here. All right. So the electrons that get re-energized here in photosystem 2, or I'm sorry, photosystem 1, they're not used. You can see that there's no exergonic process that's being demonstrated in this model. So what happens here is that those high energy electrons are actually shuttled away by NADP plus and H plus. Now, again, details like this don't matter, but just keep in mind that there are two electrons that are being carried away. One of each of those electrons sort of negate the plus symbol here and the plus symbol there, thereby resulting in NADPH, which doesn't really have a charge. So there are two electrons that are being carried away. Now, those electrons, once again, have a lot of energy. Now, before we move on to the Calvin cycle, and boy, we're running behind schedule. Yeah, that's okay. We'll get as much done as we can, and then we'll see what happens. So the electrons. So before we move on to the Calvin cycle, I want to mention something here. Why are we taking these electrons away? Well, so the idea is we're going to be building glucose from simple CO2 and H2O. If we think about glucose molecule, it's a complex molecule, as we said at the beginning of this session. And I said it requires all of these bonds. And we say all of those bonds are covalent bonds. Well, what are covalent bonds made of? Electrons. They're made from shared electrons. And these high energy electrons are required to form high energy bonds. So the light energy that go into those electrons are literally going to fuel the high energy bonds that are going to be formed in that glucose molecule. All right, so we're almost at the Calvin cycle, but there's one thing that I didn't do. Very important. If we think about what I've been saying, electrons go through this ETC and then out the ETC. Another electron goes through the ETC, out. Another one goes. If I continue to do this sort of a linear flow of electrons, what ends up happening is that you're going to run out of electrons in photosystem 2. So one of the ways that photosystem 2, or not one of the ways, the way that photosystem 2 can continue to be supplied with new electrons is through the splitting of water. And this water here gets split at photosystem 2. Remember, photosystems are very complex. They have different enzymatic regions, but there's a particular region that is capable of doing something called photolysis. Okay, some people say photolysis. I don't know. It depends on which part of America you're from, or England. Anyway, so water gets split up, one half of oxygen molecule, so one half of O2, and a couple additional protons. Wow, look at that. That actually adds to the concentration of thylakoid. So the placement of this reaction is just perfect, right? So it splits up, and then two electrons are drawn out, and then they are fed into this entire pathway. So when you give your plants water, if you guys have green thumbs, I studied plant ecology, and I'm terrible in the greenhouse. I can't grow plants. I don't know why, but I do give them water. I try to do that. And when you give them water, this is what you're doing. The electrons from water is literally going into the bonds or into this ETC and then eventually to the bonds that we see in glucose. Very important. All right. Now, Calvin cycle. Let's just go straight to the big picture here. All right. So I'm going to go through this pretty quickly because, again, the details of the Calvin cycle, not as important. So here we start with maybe around the 10 o'clock position, because in any cycle, you sort of want to start off where everything's kind of beginning. It might be an arbitrary idea, but it begins here with something called ribulose 1,5-bisphosphate. I don't know why they don't write the 1,5. Anyway, so we have the ribulose bisphosphate. It's a 5-carbon substrate. And in this case, what we see is the joining of this 5 with a 1, so that's a 1 CO2, a single carbon dioxide, via a very important enzyme that we call rubisco. Okay, rubisco. That stands for ribulose 1,5-bisphosphate carboxylase / oxygenase. And it's doing the carboxylase function by adding that CO2 to REBP. It forms a short-lived 6-carbon intermediate, which, by the way, splits into 2 3-PG. Now, I'm using those numbers, but I'm going to actually change it up a little bit. So we do have to look at this 3 at a time. 3 REBPs, 3 CO2s, 3 6-carbon intermediates, each of which splits into 2, so that you have 6 3-phosphoglycerides. Now, this is called carbon fixation. And if anyone is confused about what the word fixation means, whether we're talking about carbon fixation, nitrogen fixation, and so on, the best way to think about it is that any sort of fixation is going to take something that is non-biologically usable and turn it into something that is biologically relevant. So in this case, CO2 is inorganic. It's not biologically usable. Not in that sense. I mean, this is being used for biology, but you get what I'm saying. It's not going to be reactive biologically. And by fixing it, what we're doing is we're making it into something that is biological. 3-phosphoglycerate, this is an acid. So we actually have something that is organic. Same thing with nitrogen fixation. In nitrogen fixation, we start with something like N2, which is just the gaseous nitrogen that we have in our atmosphere. We fix it into things like nitrites and then eventually nitrates. And those are not organic per se, but they are biological nitrogen that is available for the plants. So that's what that fixation means. So we get this fixed into a 3-PG, but here's the most important thing. This 3-PG, on a scale of any other sugars that we see here or any other substrates that we see here, is relatively low energy. Okay, it's got low energy. So what happens is that we're going to start investing some energy into it from the very substrates that we created during the light reaction. So we invest ATP and we get a little bit higher energy into 1,3-bisphosphoglycerate. And then we're going to take those high energy electrons from NADPH, we invest them in here, and then we increase the energy just a little bit more into glyceraldehyde 3-phosphate. But that's really where the puck ends, right? Because G3P is sort of that apex of energy that we're going to get, as once we get those 6 G3Ps, we're going to draw 1 out. Okay, so that's 1 G3P. That's a 3-carbon sugar that comes out, and we'll see where that goes next. But we have 5 G3Ps remaining. Now, that's important because 5 times 3 is 15. And we can actually regenerate them into 3 times 5, which is also 15. So the carbons actually work out here, at least the numbers of carbons work out, and we're going to be using a few ATPs to generate REBP as well. But what's important is that this 1 G3P that comes out is basically 1/2 of a glucose molecule, because glucose is a 6-carbon substrate. This is a 3-carbon substrate. So that actually goes to show that, all right, I need to do what we see on the screen right now twice to get 2 G3Ps, and that will give me a sugar, or that will give me a glucose. But if I were to do this twice, think about how many CO2s would have to go in. So you have 3 CO2s going in now, you get 1 G3P out, but I need 2 G3Ps for glucose, so I'm going to do this one more time. I got 3 more going in. That's 6 CO2. Oh, okay, so you have 6 CO2 going in, and that yields 1 C6H12O6. That's exactly what the formula states. So this Calvin cycle here sort of verifies why the chemical reaction or the equation of photosynthesis sort of balances out the way that it does as well. All right, so that pretty much covers what we need to know for photosynthesis. But remember that things like the C4 or the CAM pathways that we see here. I don't think the details of either one are important, but I have seen questions on the AP exam where they say things like, well, this is a plant that is capable of growing in very arid environments, and you have to sort of detect that it's not going to be your standard C3 plant. So I don't think I've seen any questions like that in the recent days, so I'm going to just kind of skim over that for now. Now, I do want to hit up some questions from the previous year's exams before we move on to the other sections. There's a good chance that we're not going to be able to do like everything, but all right, so let's do this. So first, let's start with some MCQ. All right, so these are all kind of public MCQ, so you don't have to worry about this being sort of whatever, but I'm going to move my head out of the way. All right, so here we're looking at basically oxygen concentration in water. And what we see is that oxygen concentration in daylight, so presumably from 6 a.m. to 6 p.m., is increasing. And then in the dark, it's actually decreasing. So the question is, what's driving this? Now, if you know about photosynthesis, as we've been talking about, right, because we've been just doing this, we know that the oxygen is coming from the splitting of water at that photosystem too. So it is a process that contributes oxygen. And we know that in the dark, because light reaction cannot occur, we know that we're not going to be doing any of that, but probably just doing cell respiration. So we have to sort of use that photosynthesis or plants, or at least water plants, in one way or another. So A says the water becomes colder at night and thus holds more oxygen. Okay, now, this actually might be true, because I know that in colder air, there's more oxygen, if I'm not mistaken. So like car engines do better in the cold a little bit, like cold air intakes and stuff, big car guy. Anyway, it might be true, but I don't think that's something that that is going to be necessarily linked to any of the things that you talked about, right? I mean, it's sort of a red herring, isn't it? What about B? Respiration in most organisms increase at night. That is sometimes true, but most likely not. Because remember that in many organisms, assuming that they're not nocturnal, they're probably going to be sleeping. Their metabolism probably decreases at nighttime. More organisms are respiring at night than during the day. So what are they doing during the day? That doesn't make any sense. Are they holding their breath and not doing anything? That doesn't make any sense either. But D says photosynthesis produces more oxygen than is consumed by respiration during the day. So what this is actually indicating is that, yes, plants are here respiring all the time, but the net exchange of oxygen is always an efflux from the plants to the outside because they are producing more oxygen than they would consume. And what happens in light as they produce more oxygen than they consume is that the oxygen concentration goes up. And as darkness comes in, because plants are not photosynthesizing anymore, there's nothing that produces oxygen, but everything that consumes oxygen. Because plants still have to respire, and you see the oxygen concentration decreasing. So one of the things that you'll notice about these answer choices is that sometimes they're not necessarily wrong, but it's about, all right, is this going to be applicable across the grand scale of biology? Because remember, AP Bio is an intro course, right, at the college level. It has to sort of encompass the biggest of the biggest ideas, and the fact that more organisms are respiring at night during the day, are we talking about the migration of animals that come into the water, out of the water? It's like way too specific for that to be a right answer. Okay. Another one. Okay, so this one's a good one. So I told you that you don't need to know a lot about the details of the carbon of any of the light or the Calvin cycle, but this one says something like this: which of the following question is most relevant to understanding the Calvin cycle? Okay, so we know that the Calvin cycle is carbon fixation. We have the reduction. We added those ATP and NADPH. A says, how does chlorophyll capture light? No, that's the light reaction. We saw that was the light reaction. I'm going to move that up a little bit. How is ATP used in the formation of 3-carbon carbohydrates? Not bad. Not bad, because we saw that, look, we're going to invest that energy from ATP that we created in the light reaction to make that 3-PG into 1,3-bisphosphoglycerol and then 2G3P. That's good. C says, how is NADP plus reduced to NADPH? That happened at the end of the light reaction, didn't it? That's when we dumped those electrons into those electron carriers to shuttle it away. That's light reaction. How's ATP produced in chemiosmosis? That was absolutely the light reaction as well. So the thing that we see in questions like this is, well, in the bigger picture of things, how do we partition what's happening in light reaction versus what's happening in the Calvin cycle? All right, so that one is about other things. So let's take a look at a different question here. And that's going to be more about, okay, here we have something. All right. So guys, now this is going to be a challenge because I talked about the linear flow of electrons. So for example, they say in non-cyclic, so this would be the linear flow. This is the standard model. Electrons pass through the photosystem 2, and then the rest of the things, and then photosystem 1, and then it goes into NADPH. Now, some teachers will talk in great detail about the cyclical flow, the cyclic electron flow, but we didn't do that. And I didn't do it on purpose because I think there's a good chance that a lot of teachers will just kind of say, yeah, you probably won't see this, you don't have to worry about it. But take a look at this. We want to see if we can solve this by just analyzing what they're going to tell us here. In cyclic electron flow, electrons cycle through photosystem 1 and some components of the electron transport chain. So here, this is a pretty terrible model, but it's not bad. So photosystem 2, electrons get excited. It goes through the cytochrome complex and the plasticinin and all that, and that created ATP. So hopefully you guys remember that. And then it gets re-excited, and then it goes through the ferredoxin, NADP plus reductase goes into NADPH. Okay, so in the cyclical flow, what happens is that as those electrons go to ferredoxin, it goes back to cytochrome complex. So first, before we go on any further, I want to kind of talk about what the resulting consequence of this would be. All right. So in a cyclical flow, what we wouldn't get are NADPH. Because remember, this arrow is what allows electrons to leave the ETC and form NADPH. But in a cyclical flow, we don't get that. So no NADPH. But these are still high energy electrons, and they go back to cytochrome complex, which means, oh, but they can make ATP. So what the cyclical flow does is that it temporarily halts the production of NADPH, but boosts the production of ATP. Now, the reason for this really has to do with the fact that there's more ATP needed for the Calvin cycle than NADPH on a molecule per molecule basis. So sometimes cyclic flow is required for the ATP production to catch up. But the idea is, hopefully, if you're seeing this on a test and you've never heard of cyclic flow before, that's sort of where we need to be at, right? We need to see at least the consequence of what's going in and what's coming out. Now, what's going in doesn't really change, but what's coming out certainly is changing. All right. However, don't let that concern you even if you didn't get that, because you'd still get a lot of these points. Look at this. Describe the role of chlorophyll in the photosystems of plant cells. You can do this, right? Whenever they say describe, here's what you want to do. You describe it. You say the role of chlorophyll, and you have to write in full sentences, by the way, complete sentences. So you say the role of chlorophyll in the photosystems of plant cells is to absorb electromagnetic energy or light, right, of particular wavelengths. Now, you can be more specific of blues and reds in order to obtain sufficient energy to produce ATP and NADPH for the Calvin cycle, right? So the idea is that you have to talk about the capture of light energy and then being able to ready that energy for what we need to be doing with them in this entire reaction. Very easy stuff, right? Based on figure 1, explain why an increase in the ratio of NADPH to NAD+ will cause an increase in the flow of electrons through the cyclic pathway. All right, so this is actually kind of hard, okay? I think this is actually a really hard question. But the way that I need you guys to think about NADPH and NADP+ is not that they are two separate entities. NADP+ is sort of like a cup that's empty, and NADPH is a cup that's full. It's the same cup. It's just whether it's empty or whether it's full. Because here's the thing. Let's say that you have 10 cups in your kitchen, and all 10 cups are filled. Could you get any more water into additional cups? Well, no, because you don't have any more cups. All right. So when they say that NADPH is increasing relative to NADP+, what they're saying is that more of this NADP+ is being filled with electrons forming NADPH. And as that continues, the availability of NADP plus into which electrons must go is going to be diminishing. It's like I keep filling the cups that I have in the kitchen. I fill the first one, second one, third one, fourth one. I still have 6 here. 8, 9, I only have 1 cup. 10, okay, no more cups. And when you don't have any more cups, can you pour any more water into anything? No. And that's why there will be an increase in the flow of electrons that are going to not be able to go out, but it will have to go cyclical. So the way that we explain this is that we say something like, well, NADP plus is reduced by, well, it's technically not ferredoxin, but if we go by this model, say NADP plus receives electrons from the, let's say, ETC. However, once they receive electrons, they become NADPH. As more NADP plus become NADPH, there are fewer NADP plus to receive those electrons. And once those electrons cannot be received by any more NADP plus, they are then fed into the cyclic system because they cannot be exited through the end of that ETC. That's the right answer. And it's a very hard one, but that sort of justifies why the system exists in the first place. Because like I said, you need more ATP than NADPH. What that means is when the ATP runs out, NADPH is just like sitting there doing nothing. And that's exactly why the ratio actually starts to increase. All right, let's go to the application part. Using rice plants, scientists examine the effects of a mutation that results in the loss of the protein CRR6, which is part of the photosystem 1 complex. Now that's over here. And its absence reduces the activity of the photosystem 1. All right. Predict the effects of the mutation on the rate of biomass accumulation. Very easy. A lot of students see things like this and say, I've never learned about CRR4. Well, that's the point, right? That's the point of this. So biomass accumulation is about making sugars, and plants can use that sugar to, derive their own energy to build other biomolecules, or they could even store that sugar directly in things like tubers or their roots. Clearly, if you have a mutation that alters the effectiveness of the light reaction, you're going to have a bad time, right? So predict the effects, that's a prediction. So you say, let's say something like a mutation, right? Mutation of CRR6 would result in a decrease in the rate of biomass accumulation. That's C, that's it. And then D, when you justify it, you say the light reaction in which photosystem 1 is active is a very important process to produce substrates that are required for Calvin cycle. And Calvin cycle is the process through which carbon dioxide is fixed into sugars. Right? Without the ability to create sugars with this mutation, right? Without the ability to create sugars, the plants will not have the necessary energy to grow, nor have sufficient sugar to store. And as a result, they would lose biomass, right? Or they would substantially decrease in the rate of biomass accumulation. So like 3-4 sentences that connect the larger ideas. And to be completely honest, everything that you really needed to justify, as well as predict and justify what we have here, given that you know what ATP and NADPH does downstream, right, in the Calvin cycle, is all here. And it's not that bad. It's really not that bad. All right? Okay, great. So, I think I was being a little bit cavalier when I thought that we're going to be able to get through both units 3 and 4. I still have some things to talk about with cell respiration, and because I have questions on that as well. So I definitely want to get through that. But what I'm going to do for now is I'm going to put the live on pause for a minute because I think we need to get some more tea and take a break for just 5 to 10 minutes or so. So if you guys are joining us now, stay on the feed. But yeah, we'll begin again in just a few minutes. Okay, we'll be paused.
Okay, we're back. We're back. Had to get some more coffee. And about to get some more. All right, so where were we? Oh, all right. So we're moving on to cell respiration at this point. And yes, we do have to get through this in some quick succession. I know, I mean, depending on where you guys are joining us from, it might be getting very late or it might be, time to go out. It's Friday night for the American folks. So let's take a look at the cell respiratory process. So one of the things that we've been discussing over the past little while is the production of sugar and that glucose. And we said that the glucose molecule in itself contained energy as a result of those high energy bonds that are held by those highly energetic electrons. So the idea of glycolysis, as well as everything else that we're going to see here, is that the breakdown of that glucose is going to free up those electrons. It's going to break apart the bonds, it's going to release those electrons, and it's really in those highly energetic electrons that we, energize with sunlight, that we're really able to get the bulk of the energy that we're going to see here. Now, there is a little bit of a discrepancy in the textbook. Here we see these in 3 stages, but they combine pyruvate oxidation in the citric acid cycle as stage 2. But in reality, they actually put them as separate events. And I think it's a little bit better to think about it this way because there are some questions on the AP exam that really kind of pertain to this very stage, the importation of pyruvate into the mitochondria, whether that's happening or not. You might have to analyze that with the data. But let's take a look at the big picture first. So glycolysis is the very first stage. Now, I could spend a whole hour talking about glycolysis because it is a very complex process involving many, many, many different stages, but that's not really important for you guys. What is important for all the students who are taking AP Biology is where this glycolysis is taking place. Cytoplasm. Okay, so why is that important? Well, there are a few things that come to mind. One is that glycolysis is commonly referred to as a universal process in biology. And whenever we use the word universal, it really means that I'm going to take that back. Let me walk it back. When we use the word universal in biology, that means that every single living thing has the capacity to do glycolysis. Now, when I say almost, what I meant was that some of them may have evolved to not do glycolysis, but the initial state was that they should be capable of doing glycolysis prior to the adaptation. So it's a universal process. So how does that link up with the idea that it occurs in the cytosol? Well, because when we think about the word universal, that would include eukaryotic cells, but it would also include all the prokaryotic cells, whether it be bacteria or archaea. And one of the main distinguishing features of eukaryotes from prokaryotes is that prokaryotes don't have membrane-bound organelles. So if it's a universal process that occurs in all living things, it makes no sense for it to take place in an organelle that some other or some subset of those living things do not have. So it occurs in a common area that everyone has, which would be the cytoplasm. Now, another very important factor is that glycolysis is anaerobic in nature. Now, I know that this whole aerobic cell respiration, as we see in this entire image, does require oxygen, but oxygen is really right here at the very end. It's called a final electron acceptor in oxidative phosphorylation. If we look at glycolysis in isolation, it's an anaerobic process, and it's also very ancient. So now we have to link those ideas with this idea of universality. So once again, if we think about something being universal across all taxa of living things, be it prokaryotes or eukaryotes, it means that it must have evolved in the last universal common ancestor, right? It evolved a long time ago such that it's now a homologous trait that we receive from that common ancestry, which would mean that it would have had to evolve a very long time ago. And that's sort of the ancient idea. And that also links up with the fact that it's anaerobic because in the early days of our planet, we didn't really have a lot of oxygen in the atmosphere. In fact, we had a bunch of like the great oxidation events, with the evolution of photosynthesis and so on in the land plants. So a long time ago, we didn't have a lot of oxygen. So it makes no sense for glycolysis to be aerobic in its, isolated nature. So those are some major ideas that AP wants you to kind of connect to glycolysis. So with that being said, let's talk about what happens in the macroscopic picture. So glycolysis comes from the word glycolysis. So that would mean that we're cutting sugar. And it cuts that 6-carbon sugar into 2 equal
acids that we call pyruvate. So that would be pyruvic acids. So these are each 3 carbon. It gets fed into the mitochondria and then manipulated through the citric acid cycle. And during this process, all of those carbons from pyruvate are released as carbon dioxide. So that's sort of where that CO2 on the product side of cell respiration is going to come into play. And then you have the electrons that are being stripped away, which are going to be run through the oxidative phosphorylation. There's electrons being stripped away in glycolysis as well. Let's take a look at glycolysis first. And really, we just need to know this big picture stuff. Okay, so it says glucose going in. There is an energy investment phase, 2 ATPs being used. We get 4 ATPs back, so that's a net gain of 2 ATP. You don't really have to worry about water here. But what's important is that when we cut that glucose in half, we actually get 4 electrons that are released. Now, these are those high energy electrons that we've been talking about for the last hour and a half. Those high energy electrons are going to be carried away by NADH. Now, I know that this can be a little confusing because in photosynthesis, we learned about NADPH. Here, we're learning about NADH. Now, not that this is why the naming's different, but you can think of that P as indicating like photosynthesis. Like NADPH is the one in photosynthesis. In the absence of that P, we have NADH, which is in cell respiration. So remember I said that each of those NAD plus molecules can receive 2 electrons. Because we have 4 electrons, we're going to get 2 NADHs being produced here. And as we said, we get 2 equal part pyruvic acids or pyruvates at the end of this entire process. So that's basically glycolysis in a nutshell. Now, I think one thing that you guys need to keep in mind is that as we talk about anaerobic cell respiration at some point in the future, what we see is that the anaerobic respiration is simply trying to continue glycolysis without oxygen. Now, I know that I said, well, glycolysis is not an aerobic process. Well, that's because this NADH that's produced is going to have to return as NAD+, and this regeneration part is where you need oxygen. So that's sort of how that's going to link up later. Okay, so let's take a look at what pyruvate looks like, because I think this picture is great. And this is, again, another reason that I think looking at pyruvate oxidation as a separate step is so important, because you really do get a clear sense of what exactly is happening to that pyruvate, which is 1/2 of that glucose molecule. So as we mentioned, pyruvates are 3 carbon substrates. And look at what is grayed out here. That's actually CO2. And during the transport of pyruvate into the mitochondria, that CO2 actually gets released. So that is that very first CO2 that is oxidized off of what was formerly a sugar that gets returned to the environment. And this is the CO2 that we breathe out every time we take a breath. So once that CO2 leaves, these 2 carbons are going to be there. But a couple of things. In order for us to release that CO2, what are we breaking? We're breaking that bond. That bond is made from electrons. How many? Well, a single bond in chemistry is said to be a sharing of 2 electrons, a pair of electrons. So 2 electrons come out. Well, 2 electrons interact and gets captured by NAD+ and an H, and then you get an NADH. So that NADH right there is carrying that high energy electron right here that's been stripped away when that CO2 was released. The second part is that these 2 carbons forming the 2 carbon substrate is sort of unstable. So it sort of interacts almost instantaneously with coenzyme A. And you can see that coenzyme A almost like a hat, right? It's like a cap that gets put on top of those 2 carbons to stabilize it. And this is what we call acetyl-CoA, all right? Now, remember that if we're accounting for glucose, each glucose molecule would produce 2 pyruvates. So by the time both pyruvates go in, we would have released 2 CO2, we would have formed 2 NADH, and we would actually have 2 acetyl-CoA. Great. Now, that acetyl-CoA goes into the citric acid cycle. And by the way, if you guys are studying from the textbook and this picture confuses you, this right here is just a simplified version of what we see here. So this is not another thing that's happening. It's just being summarized. This is pyruvate oxidation. We get that acetyl-CoA, and here we go. Acetyl-CoA. Now, this picture is, I would argue, almost sufficient in what you need to know for AP Bio. Okay, you don't need to know every single one of the substrates and the enzymes. Of course, the enzymes are not even mentioned here, but this picture is, I think, good enough. And let me explain what's happening here. So the acetyl-CoA contains 2 carbons. Coenzyme A is actually released as acetyl-CoA enters the citric acid cycle. it goes through a series of reactions, kind of like what we see in Calvin cycle. But instead of the Calvin cycle for the citric acid, we're sort of going the other way. Because instead of capturing carbon, we start releasing carbon. We have those two carbons that we've added in leaving. And that makes sense because by the end of the cycle, everything in terms of the atoms should be sort of the same. So those two carbons get released as CO2. We get additional electrons here, 6 electrons that are being captured by 3 NADH, another 2 electrons being captured by FAD, which is another electron carrier. It's sort of like saying, look, we have a Ford F-150 and Chevy Silverado. Okay, so for Americans out there, I grew up in Ohio. Haha. Okay, anyway, but the point is, they're basically the same thing. They're carrying those electrons away, and you also have this ATP forming as well. All right, so this is sort of a complicated process. Now, I'm going to show you the big picture. All this is saying is that as we release CO2, electrons are free, we get NADH. Release CO2, electrons are free, we get NADH. We have this sort of a conveyor belt system that allows us to phosphorylate ATP. We have additional weird things that we're doing with double bonding and relieving double bonds, but ultimately 3 NADH, 1 FADH2, 1 ATP per acetyl-CoA. Now, once again, if we're doing this per glucose, you guys know you'd have to double all of this. You get 6 NADH, 2 FADH2s, and 2 ATP. But why this is important is that as we get to sort of, is there another? Okay, maybe there's a big summary picture that I want to show you. Yeah, as we get to this picture, we've created 2 ATPs thus far from glycolysis, and we create 2 ATPs from citric acid cycle. We said that because, again, 2 acetyl-CoAs going in, each of which produces 1 ATP. This is all being accounted for per glucose. And we know that at the end of the day, we're supposed to get 30 to 32 ATPs. So we've only made 4. So there's this remaining 26 to 28 ATPs that have not been accounted for. And all of that energy required for that ATP production is held within the NADH and FADH2s that we've been producing during glycolysis, as well as pyruvate oxidation and citric acid cycle. And all of that gets fed in to this oxidative phosphorylation. So let's take a look at what that looks like. So just to put things into perspective, we saw that pyruvates were being imported into the mitochondria. Hopefully by this point, you guys are familiar with the double membrane structure of the mitochondria. And this right here, the phospholipid bilayer that we're seeing, is the inner membrane. So that's the inside. And what you don't see is the outer membrane. But the outer membrane is here. There is an outer membrane. And this dark orange area is the intermembrane space. And that's really good because here's what we're going to do. We're going to bring some of those NADH and FADH2s. Now, along that inner mitochondrial membrane, we have the electron transport chain, very reminiscent of what we saw in the light reaction. But instead of using electrons that are energized by light, we're actually starting off with electrons that are already highly energetic because they are electrons coming from glucose. And glucose bonds were very powerful, right? They were very strong. They have a lot of energy. So those electrons come in, and from NADH, it enters the system at the very beginning. So let's follow those electrons. It goes through protein complex 1 and converges with other electrons at ubiquinone, known as Q. It goes through protein complex 3, cytochrome C, and eventually to protein complex 4. But during this process, that energy of the electron that starts off high is actually being used. Once again, you can think of the electron transport chain here, and as well as what we saw in the light reaction, as sort of this system that can draw out the excitation energy of the electron in a controlled manner. And this, again, is being used to do something called pumping of those protons. But in this particular case, we're going to pump them from the matrix of the mitochondrion into the intermembrane space. Now, remember I said it was good that there was an outer membrane, because as we pump these protons, instead of just diffusing away from the mitochondria, because we have an outer membrane, it gets packed into this intermembrane space. It really gets highly, highly concentrated there. And just as we discussed this in the same light, no pun intended, in the light reaction, these protons are going to want to fall back in. They can't do that through the lipid bilayer because the lipid bilayer is nonpolar, so they come in through the ATP synthase, and as they do that, once again, you're going to make ATP, all right? Now, for FADH2, they do enter this entire system a little bit later, and they do so at protein complex 2, and again they converge at Q. But notice the difference. When NADH enters earlier, they have this extra piece that allows for additional protons to be pumped, whereas FADH2 converges with the NADH electrons and starts pumping at 3, which means that when we do the calculations of the impact of each one of these electron carriers on the buildup of that concentration proton gradient, well, FADH2 produces slightly less ATP or slightly fewer ATP than NADH on account of the fact that it starts off a little bit later, pumping fewer protons into the entire mix. Now, this isn't because the electrons in FADH2 are weaker by any means. It just means that the whole linear system that we're dealing with is sort of designed in this way through the course of evolution, of course. Now, let's take a look at what else happens here. The electrons that are arriving at protein complex 4 must be relieved. Otherwise, you're going to get a backup of all those electrons. As in, if you don't relieve those electrons out here, you can't add any more electrons to protein complex 4. So cytochrome C gets backed up, and then protein complex 3 gets backed up, and this sort of creates a problem. So how do you get rid of those electrons? Well, you can get rid of those electrons by using a chemical, using an element that is really good at ripping away electrons. That is to say, really good at oxidizing. And the name is oxygen. That's precisely why oxygen is used in cell respiration. It's very good at picking up electrons, and what it does is that at the end of this ETC, it takes away those electrons, combines with protons to make water. Okay, so it's called the final electron acceptor. It takes that water. By the way, this looks like the inverse of photolysis that we saw in photosystem 2. That's exactly right. That's what's happening. So the electrons are captured, which then allows for a free flow of electrons through additional input of NADH and FADH2. Now, what's important also is that as NADH drops off those electrons, as FADH2 drops off those electrons, we kind of talked about this with NADPH as well, but it's sort of like a cup being emptied. So that allows for this empty cup, NAD+, and this empty cup, FAD, to return to the Calvin cycle to pick up more electrons from the breakdown of food. And the reason that this is such a critical point is that if oxygen is not available, as we say, if you're in an anaerobic condition and oxygen is insufficient or unavailable, you'll notice that NADH nor FADH2 can drop off their electrons anymore because the electrons are all backed up. So the cup is not being emptied. No NAD+, no FAD, no empty truck beds. They're all filled, so you can't put any more electrons into those electron carriers. What does that mean? That means the citric acid cycle cannot proceed because the citric acid cycle needs those empty cups right there. It needs that empty cup. It needs that empty cup. It needs this empty cup or the flatbed, however you want to think about it. Those electron carriers are not available anymore. And as such, the citric acid cycle stops. And when the citric acid cycle stops, acetyl-CoA can no longer drop off these 2 carbons. So coenzyme A is now freed. The coenzyme A is stuck with acetyl-CoA. Now, why is that a problem? Well, because it turns out that you need coenzyme A to bring in pyruvate into the mitochondria. So, without oxygen, the mitochondria closes down. And that's why we have glycolysis as the only means of producing ATP when oxygen is unavailable. But even that glycolysis produces a problem. NADH. This is supposed to go all the way into oxidative phosphorylation to return as those empty cups. So what organisms have evolved is a way that in the cytoplasm that we can pretty much regenerate that NAD plus by basically dumping those electrons that are in NADH into whatever substrates that we can create from pyruvate. Now, the rationale is actually pretty simple. Think about it this way. Let's look at alcohol fermentation, right? In the absence of oxygen, this pyruvate is not going to do much for us because it cannot go into the mitochondria. And as such, and again, yeast cells are going to do this, so they have mitochondria, but mitochondria, they can't do it. So they say, well, who cares then? I don't really care about this pyruvate anymore. I can't really break it down anymore. So what I'll do is I'll turn this into acetaldehyde, and then once it's in this acetaldehyde form, then I will dump those electrons from NADH into that to create ethanol, and at the least, I'll be able to get this NAD plus back. I'll be able to get that empty cup back, so I can just do that glycolysis, at least 2 ATPs per turn, right? Because that's at least going to keep us afloat while oxygen becomes available. Now, for yeasts, 2 ATPs per glucose, not the end of the world. They don't have a very complex, demand for energy. But what about animals? Well, animals can do this too. For us, it's actually a little bit simpler. We say, look, we're going to take that pyruvate. We're just going to dump those electrons from NADH right into pyruvate, producing lactic acid. Now, lactic acid, again, is an acid. It's not great. Sometimes we have this misconception that this is what causes muscle soreness after you work out. It turns out that it's a lot more complicated than just lactic acid, because once lactic acid forms, it basically goes to the liver and gets regenerated into pyruvate and glucose, so it can kind of do this whole thing again. But the idea is, for animals, this may be insufficient, as in we can't really survive on just doing lactic acid fermentation, if we look at humans. So the purpose of lactic acid fermentation is in the context of all of the cells that you have in your body. Let's say you're doing a sprint, you have a lot of muscle cells. Some of those muscle cells are going to be doing lactic acid fermentation as they await for availability of oxygen that is sort of insufficiently being brought by the limited vascular system that we have, the limited veins and arteries that we have. So at least they serve a function in those temporary regards. Okay, so that is basically going to do it for cell respiration. Now, I think that will give us an opportunity to look at some of these questions. So let's go back and take a look at maybe this one. This one's good. So I think this is from a relatively recent FRQ, and it says something like this. To investigate how increases in environmental temperatures affect the metabolism of some organisms, researchers incubated liver cells from toads at different temperatures and measured two markers of metabolic activity. The rate of oxygen consumption, great because oxygen is the final electron acceptor in aerobic cell respiration, and rate of ATP synthesis, which is happening like right next to that. It's all happening in mitochondria. So like, again, as I said, you don't need to memorize the entire Krebs cycle, but like know where these things are taking place. All right. A, describe the role. Oh, by the way, this is question number 2. So this is the big one, right? All right. Describe the role of water in the hydrolysis of ATP. So in the new versions of this test, these questions are always very predictable. The first A is always going to be some general background knowledge. So what is the role of water in the hydrolysis of ATP? So this is something that we've mentioned before. ATP, which is a complex substrate. I'm going to try to find a picture of this, actually. It's this picture right here. So ATP, which is a relatively complex substrate, is created from covalent bonds, right? Whenever covalent bonds are broken, we use water in performing something called hydrolysis. And through hydrolysis, complex materials can be broken down into simpler substances. That's basically all you have to say, right? AP is not expecting you to say, well, there's like a nucleophilic attack of electron. Actually, I don't even know if that's relevant. But the point is, you don't need to go into even like the H goes to one side, OH goes to the other side. Even though we've seen that in hydrolysis diagrams from Unit 1, I don't even think you need to do that. That's very easy stuff. Now let's look at the graph. As temperature goes up, rate of oxygen consumption goes up, so respiration seems to increase. Rate of ATP synthesis also seems to increase. Now, one thing that you guys might not have picked up on is that this is supposed to be like toads. So I guess toads they have greater respiratory rates when temperatures go up. I think they might be like are toads ectothermic? Hold on, I'm going to look this up. Oh, I should probably type in toads. Hold on, I know you guys can't see this right now. Yeah, toads are ectothermic. That's right. Okay, so that means that they actually have, they don't regulate their internal temperature, right? So as the temperature goes up outside, they just do more. Well, that makes sense because like lizards are ectothermic and they're actually not even amphibians. They're actually evolved past that. Okay, constructing a bar graph. Now, I'm glad that we're seeing this because they underlined this for you. I can't tell you how many times students have come back and said, yeah, I drew the wrong graph. I drew a line graph. And like, dude, it says to draw a bar graph. Now, in the past, I want to say just like even as far back as like 6 years ago, they would give you data and they wouldn't even tell you what kind of graph to use. So you have to kind of figure it out. And I'm sure you guys have done some of those exercises at school where you say, oh, well, if the x-axis is going to be a continuous variable, then use a scatter plot and so on. In this case, you actually do have a continuous variable on the x-axis, right? Because the metabolic marker here, these are going to be our bars, right? These are going to be our bars with the plus and minuses. This forms our x-axis. And what we actually see is that they tell us to use a bar graph. And I think in some ways that is sort of emblematic of them saying, like, I know if we don't tell you to do a bar graph, your teacher probably would tell you to do a scatter plot, and that is technically okay too. Anyway, so you can graph that. And remember, the main important thing here is that the rate of oxygen consumption, oh, did they tell you? Okay, so all of the data. All right, the rate of oxygen consumption and the rate of ATP synthesis, okay, you have to actually represent both of them in the graph. So what I would say is that the x-axis will be labeled as temperature, and then you would write centigrade or Celsius. You have 20, 25, and 30 as these discrete points at which bars are going to come out. Metabolic marker can go on the y-axis, and what you can do is use rate of oxygen consumption and rate of ATP syntheses as sort of differently shaded bars that are all next to each other. So you have 12.8 and 12.6 right next to each other within the 20 degrees. And what's really great about what we have here is that the nanomoles per mil per milligram, nanomoles per mil per milligram, this is the same, right? This could be very, very challenging if these were different, because then you would have to have two y-axes with like two very different measures. Like imagine if this is, 4,000, right? How are you going to draw 12 and 4,000 right next to each other, right? The only way that you would do that is to have this measure on a separate y-axis so that you can try to get everything into a single picture. But thankfully, they're not going to make you do that. These are all going to be relatively similar data points with the same unit. All right, determine the temperature in Celsius at which the rate of oxygen consumption is different from the rate of oxygen consumption at 25 degrees. What do they mean when they say different? This is the toughest thing, because I feel like this is one of those like very AP bio skill sets that, most teachers do teach, but some students forget or they're not paying attention. Here's what it is. Anytime we're using the word different in science, it actually means something. It's not just about means. It's not just about the fact that 22.1 is different from 21.07. What it's asking is it significantly different? Is it statistically different? And we're using 25 degrees. I think they're saying oxygen consumption at 25 degrees. So this is right here. This is the data that we're working with. What we see when you graph this are error bars that actually extend above 16.5 to 18.5 and below 16.5 to 14.5. And the idea is that if they're different, then the error bars of the other temperatures would not overlap with the error bars of the 25 degrees condition. So we can do this sort of in our heads too, because we know that 25 degrees yields greater oxygen consumption than 20. I can sort of look at the lower end of that spectrum. The lower end of that spectrum is 14.5. But the upper end of the 20 degree spectrum is going to be 15. 14.5 and 15 overlap. So these are not different. However, let's take a look at going the other way. So because 30 degrees yields a higher rate of oxygen consumption, I'm going to look at the upper end here. It's going to go up to 18.5. Now, 22.1 minus 0.7 gets us something like 21.4. 21.4 is yet higher than 18.5. So these are actually different. These are not. These are. So when you do this question, you have to say the temperature at which rate of oxygen consumption is different than the rate of oxygen consumption at 25 degrees is 30 degrees. That's what you do, is 30 degrees. That's it. Very easy. Like so far, we didn't even need to know anything. Like hydrolysis, that's unit 1. Drawing graphs, interpreting differences in data. Half of this question, by the way, this is number 2, right? Didn't even require biology. Like you could do this if you just know some of these skills. Now, here's the next skill that comes into play, all right? The next skill is that AP Bio wants to test your ability to write lab reports. Now, what do I mean by this? Well, AP Bio, as you guys know, is offered as a laboratory science class. And depending on what school you go to, you'll do labs. In some other schools, you won't do labs. If you're studying this on your own, you're definitely not doing labs, unless you're very, very keen. But we learn about how to write lab reports. And on the lab report, if you've done it, you have things like introduction, you have like materials and methods, and then you have this thing called results. And in the results section, what you do is you present your tables, you present your graphs, and then you sort of explain. what those graphs or tables are saying. That's the test in number 2C. It's almost always 2C, right? They always say, based on this data, describe the effect of temperature on the rate of ATP synthesis. That's what you do in a lab report. If you collected this data, this is what you do. You say, all right, well, ATP synthesis has a general trend of increasing as temperature increases. We see this with 12.6 plus or minus 1.6 at 20 degrees, rising up to 16.8 at 25 degrees, and as far as 21.07, and each of these increases are statistically significant. Now, do you need to say all of what I just said? No, but if you did, and you have time to, that is the perfect answer, because that's what you would write in a lab report. That's the skill test that they're doing. And then on a lab report too, they might say, well, there's a math section, right? Some teachers will say, oh, you got to put the mathematical equations that you use. Well, they're going to actually ask you to do some calculations. Average amount of oxygen consumed for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees. This seems hard, but it's not. Okay, let's take a look at this. Calculate the average amount of oxygen consumed. So we're looking at the top, and they're saying 25, so we're looking at this number right here. And they're saying for 10 milligrams for 10 minutes. Wait, hold up. How do I do that? I don't have milligrams and minutes here, but I do here, because what this number represents, and by the way, by the time you take AP Bio, I know you guys are pretty good at math. There's always an implicit 1 in front of all these, right? It's 1 nanomole per 1 minute per 1 milligram. You got to know that, because if it's saying, look, at 25 degrees, we're producing 16.5 nanomoles of oxygen, consuming 16.5 nanomoles per minute per milligram, then you ask yourself, well, then how much would we get in 10 minutes? I don't know, you multiply that by 10. Okay, but that's per milligram. What about for 10 milligrams? Then you multiply that by 10 too. So you basically have a 10 and a 10. You multiply the 16.5 by 100, and that's your answer. That's it. Very easy. So again, so far, this looks like a Unit 3 question. And I've been just talking my head off, reviewing all of this, detail, but you don't even need to know any of that. I mean, you might need to know that for MCQ, but like right now, we didn't need to know anything. This is not a biology test. It's crazy. All right, anyway, D. All right, maybe we have something here. Let's take a look. Oligomycin. That sounds like an antibiotic to me. Oligomycin is a compound that can block the channel protein function of ATP synthase. All right, we got to know that, right? We got to know ATP synthase. And even if you didn't know ATP synthase, look at the name of the enzyme, right? And if someone said, hey, what does ATP synthase do? You say, I don't know, it probably synthesizes ATP. All right, fine, but it's blocking it. So I'm guessing, again, even if you don't know what it does, it's blocking it. That's not good. It's not going to let it do what it does. Predict the effects of using oligomycin on the proton gradient across the inner mitochondrial membrane. Okay, now we actually have to know stuff. All right, fine. Let's do it. We're going to go back to that picture right here. Oh, that's too zoomed in. Right here. Okay. What this question is about is that very important idea. Remember earlier I said, this time we're going to pump protons into the intermembrane space. And it's really important because it can be compacted there. We're taking it from the matrix. We're going to pump it to the intermembrane space. Now, they fall back through the ATP synthase. But we're going to block this ATP synthase. I think that much is pretty clear. We block this. So what happens? What happens to the concentration gradient? Now, whenever they say concentration gradient, if you don't know what that means, that's going to be troubling. But it's really about the difference in this proton concentration here versus here. So if the ATP synthase isn't working, but presumably everything else is, you're going to have that concentration gradient get bigger and bigger and bigger and bigger. You're going to have a greater differential between protons on the outside and the inside as we continue this. So let's do that. Prediction and justification. If oligomycin is used, then the proton gradient across the inner mitochondrial membrane will increase. That's it. That's the first part. Okay, part 2. This is because ATP synthase is used to allow those protons to return into the mitochondrial matrix to form ATP. But if it's not returning into matrix, but there's still all of the other ETC functions that are pumping protons into the intermembrane space, you're going to get that concentration gradient increasing. Okay, now what else could they have done? They could have said, predict the effect of using oligomycin on the pH of the intermembrane space. All right, So we talked about chemistry a little bit earlier. I said all you need to know for pH is that you have more protons. You have more protons there. So you would say, well, it's going to be more acidic, and it's also going to be a lower pH, right? You see how they can play around with this. It doesn't have to be always about the proton gradient. Whenever it's protons, they can always kick it up a notch and say, all right, well, let's test our AP students' abilities to detect that this is going to be a pH issue, right? You can do that too. But it's really easy, right? Because in order to do that, in order to solve by the way, let me just walk this back, all right? In order to solve number 2, you just need to know very rudimentary things about hydrolysis, which is very important. I mean, if you don't know that, that's not a detail, right? That's a very important thing. And knowing how to draw graphs, knowing how to read tables, that covers almost 8 out of 10 points. Now remember this, guys. AP Biology, in order to get a 5, it depends on the curve. And by the way, the curve is not just how you guys solve it. The curve is predetermined. It's about getting about 77% to 80% on the overall scoring. And what that means is out of 60 questions, you want to get about 80%. And on FRQ, out of 60 points, you want to get about 80%, or just any combination that's going to put you, if you get 80%, you're kind of, in the right area for a 5. It's on the lower end, but you can still probably guarantee that you'll get a 5. Now, for this question in number 2, what we see clearly is that, you didn't really need to know a lot, and this is definitely from energetics, but you could probably get away with getting an 8 out of 10 for this question, because you can do A, you can do B, you can do C. You might not get D. Each of these are usually 1 point, 1 point. You get 8 out of 10. That's not bad, right? That's not bad. That's going to get you in the right area. Let's take a look at another one. This is also a number 2, okay? Elevated levels of CO2 increase the rate of photosynthesis and growth in plants. That makes sense. Scientists studying and the mechanisms involved in these increases examined a variety of species and found that when plants are exposed to higher CO2, there's an increasing number of chloroplasts. So that makes sense. They're responding to greater amount of CO2 by producing more chloroplasts, or allowing chloroplasts to proliferate more to take advantage of it. So we want to study whether these elevated levels of CO2 have similar effects on the numbers of mitochondria. And then they took 6 of these species to quantify the number of mitochondria per cell when plants were exposed to both normal and elevated CO2 levels. So mitochondria and normal, we have basically, these are averages of each species. So you have 1 and you have 0.4, blah, blah, blah. And then you see elevated CO2. I think what we see is a general trend towards increase. And again, if we were to take a closer look at these errors, right? So the higher end of the normal CO2 for species 4 would be 0.33. The lower end here would be 0.54. So these error bars would technically not overlap. So that's actually good. All right, look at this. Again, A is about the general biology. Describe the role of inner mitochondrial membrane in cell respiration. So I'm not going to do that again. This is going to be where the ETC is housed. This is where you have ATP synthase. You also have this inner mitochondrial membrane, which allows for a concentration gradient to be held as well. So that's, again, very general information, constructing the graph. Now, in this case, they actually don't tell you whether this should be a bar graph or anything like that. But the way that you should be approaching this, of course, is that if you have, let's say, normal versus elevated, there are 2 different ways that this graph could be created, right? You could have normal versus, I don't know if you can see the mouse, but normal versus elevated, or you could have species 1, 2, 3, 4, 5. I would prefer to have the species along the x-axis so that you can look at each of the species and see how that difference in normal versus elevated CO2 would be in terms of the bars. That's one of the ways that you can do it. And then determine which species shows a difference. Once again, it's the same thing, right? 2 different years, same dang question. That difference means something, as I said. So we're going to be looking for significant differences. And I'd have to actually look at this in slightly greater detail, but it does look like almost every one of them is going to be pretty different between normal and elevated. Describe the relationship between the level of CO2 and the average number of mitochondria per unit area of the cell. Once again, you can do that because this is the sort of the results section of a lab report. All you're doing is you're describing data. Very, very easy stuff. Lastly, the leaves of a particular plant species are typically green, but scientists notice a plant in which the leaves have white stripes. They determined that the stripes result from a mutation in mitochondrial DNA that interferes with the development of chloroplasts. Well, that's interesting. I don't know if that's real, right? I don't know to what extent AP biology like makes things up. But anyway, the scientists cross plants using pollen. Oh, there it is. I talked about that earlier. Look, I don't know how many of you guys are joining us now, but if you've been here since like 2 hours ago, I've been talking about how AP Bio might do this really sly thing where they talk about mitochondrial inheritance, but use words like pollen. Look at this, pollen from plants with white striped leaves and ovules from plants with green leaves. But this is a mitochondrial problem, and only the female plants that produce the ovules are giving the mitochondria. So predict the phenotypes of the leaves of the offspring of this cross. They'll all be normal. They'll all be green because the pollen does not contribute this mitochondrial issue to the offspring. Look at that, right? They do this kind of stuff. All right. Provide reasoning to justify your prediction. I just did that. Explain why plants with the same genotype are able to differ in structures or number of certain organelles in response to changes in atmospheric levels of CO2. Okay, so this one is a little bit different. So let's partition this last part because this is not really about that whole mitochondrial thing. Whenever AP Bio is asking, how is it that same genotype are able to be different in whatever regards that we're talking about? I hope you guys know this. That's all gene-environment interactions, right? It's about the extent to which environments can alter the expression patterns of genes, which is then related to the regulation of gene expression. But the extent to which you need to investigate the details of that in this particular case is not very high. You don't need to go into very much detail. All you have to say is that some plants with same genotypes are able to differ in their structure or number of organelles because the environment has an impact on the level of gene expression, and the regulatory pathways can result in greater or fewer number of organelles being produced or dictated by the nucleus. Right? You didn't talk about mitochondria or chloroplast because, how are you supposed to know that? Right? This is not Cytology 350 or plant cell biology in like Stanford University. Like you're not privy to that information, and you're not supposed to be. All you say is, look, that's the depth at which we're going to be traveling. We're going to say there's environmental factors in gene expression regulation, and those regulatory pathways will result in differential organelle production, blah, blah, blah. And again, if you want to go really deep, like the deepest you can go is which provided an adaptive advantage for organisms who could do that, right? Like that's basically it. It's it's not bad, right? So I look, my hope is that as we talk about these types of questions, you start to see, hey, you know what? Like this is actually not bad. Like I know this stuff because I think part of the problem with, look, if you're watching this right now or later, doesn't matter. But if you're watching this video, chances are you care about AP biology. You care about your grades. So that usually means that you're going to go into the test and you're going to actually have studied pretty hard. You know what you're doing. The problem is to connect what you know to what they're asking. And that's what I'm hoping to do. So let's do one more. And then I think we'll have to end the broadcast for today. I'll have to just kind of like pick up on Unit 4 later. I think I was being crazy to think that we can do 2 units in one day. It's just I go on these tangents and I blab a lot, right? And also, this is wonderful. There are a few viewers right now, and I hope you guys know that you're actually making my dream come true because, I'm Korean, so I thought I'd be a StarCraft streamer at one point. But it turned out that I'm pretty bad at StarCraft, but I'm better at biology. So this is fine. This is fine. All right. So Blizzard, if you're watching, I can play a little bit, or at least I can like talk about games and stuff. Anyway, let's go. Fireflies emit light when the enzyme luciferase catalyzes a reaction in which its substrate D-luciferin reacts to form oxyluciferin and other products. So that's what we have in Figure 1. So in order to determine the optimal temperature for this enzyme, scientists added ATP to a solution containing D-luciferin, luciferase, and other substances needed for this reaction. They then measure the amount of light emitted during the first 3 seconds of the reaction when it was carried out at different temperatures. Now, guys, I want you to take a look. This is number 3. So notice how they're not giving you any data tables. This is not supposed to be one of those questions. It's more sort of a theoretical question, okay? So we have all the substrates here: D-luciferin, oxygen, ATP. You have this luciferase produces oxyluciferin, CO2, AMP, and that's a pyrophosphate. That's 2 phosphates because remember, ATP is 3 phosphates here, mono is 1, and there's 2, and there's 3, and then there's light. So the reaction is relatively balanced. Okay, so like, do I need to any of that? I don't know. Again, this is AP Bio, so I might not need to know any of this. Describe a characteristic of luciferase enzyme that allows it to catalyze this reaction. Yeah, we totally did this. Okay. Chances are, if you're watching this right now, you weren't here at the very beginning. So look, let me put it this way. Luciferase enzyme, or whatever enzyme it is, has a particular structure that is encoded by the polypeptide structure or the amino acid sequence. This structure allows it to bind to a particular substrate, in this case, bind to D-luciferin, which then through the induced fit model, which by manipulating the activation energy, it allows for this reaction to be catalyzed. That's it. Once again, you can't go any deeper than that because you don't know luciferin. We don't know luciferase. I don't even know what it looks like. All I know is that it's an enzyme, and this is the way that enzymes always work. Okay. And then we have some of these experimental questions. What is the dependent variable? Do they tell us? So they're going to like use different temperatures, measure the amount of light. Oh, there it is. Amount of light emitted during the first 3 seconds of the reaction. So you literally copy and paste this, right? I mean, I know you can't. You have to write this down. But the dependent variable in this experiment was the amount of light emitted during the first 3 seconds of the reaction. All right, there we go. It's AP by FRQ. Like you're going to be repeating pretty much 40% of what's already written, and then the other 60% is what you write out of your noodles. Okay, good. State the null hypothesis. All right, so this one's a little bit tricky. Remember the null hypothesis for any experiment. Think about the word null. It means that what you're predicting to have an impact is not having an impact. So the prediction is that there's going to be an optimal temperature, and presumably that means there are going to be temperatures at which this enzyme is not operating properly. So all you got to say here is that temperature has no impact on the reaction rate performed by luciferase. That's it. That's one of the ways you can do it. Like you don't have to be like from low temperature to high temperature. No. Just say temperature has no impact on the luciferase's ability to carry out this reaction. That's another way to put it. That's it. So far, so good. Again, not a lot of real knowledge, except for this one, of course. D, this is always the one. A student claims that as temperature increases, there will be an increase in the amount of light given off by the reaction in the first 3 seconds. Support the students. Oh, that's easy too. So yeah, we really don't need to know anything about oxygen or anything. Okay, so that's good. We know this. Okay, the speculation here is that look, you're supposed to support it. So you please don't go the other way and be like, well, as temperature increases like a lot, the enzyme will denature, and then the student's claim is dumb because it's not going to. Don't do that, right? Because they're asking you to support this claim. So then reel it back a little bit, because the students who know this a little too well have argued with me and said, oh, but it's not true. I was like, yes, it's true, but we don't know how much this increase is going to be. So then now we're going back to just that general idea of increased temperature resulting in an increased level of kinetic energy, which just speeds everything up. We know that. We know that warmer temperatures means greater vibrations of molecules, greater bombardment, greater collision. More collisions simply means more reactions. And that's all you have to do. So this is not even bio. That's like, I don't know, if you guys are taking general chemistry or AP Chem or even like physics, you can solve that. Right? I can give this to my physics class, and I don't teach physics, but I can give it to a physics class, and they should be able to do that too. Very, very simple stuff. And these were questions like, number 4, we talked about that already. Number 3, number 2, this one's number 2. Like, these are the big questions. These aren't even this, like the number 7s and 8s, although 7s and 8s are, equally as hard as 3, 4. So I'm sorry, there's no 7s and 8s. It's 3, 4, 5, 6, right? They're all pretty much even, but it really, guys, AP Bio FRQ it's not hard because it's not that detailed. So the 2 things that you have to understand is by having the sense of knowing what they're trying to ask and what the big themes are. That's one. And then as you explain that, understanding the limits of how deep you can go. Like even if you've done a summer research program at Emory or Chicago and you're like, oh, actually, I do know this very well. Don't go there. You know what I mean? You don't have to go there. You have to understand. Like even if you're an Olympic swimmer, like you can still swim at a moderate pace, right? You just got to get to that moderate pace. And you have to understand what that moderate pace is in order to do well on this test. So, guys, that will do it for today. I presume that YouTube will just kind of automatically upload this to the channel once it's done. I have no idea how this works, but it's my first YouTube live. I'm going to, I'll be posting on our community so that you guys can be on top of when we'll do the next meeting. If you're joining us from our Discord server, then I'll be also posting the next meeting as well. I know we're running out of time. We only have about a month, but listen, I have another video coming out next week. That's going to be a 50-minute video that covers everything from units 1 through 8, and not this detailed as we did today, but all the main themes. So be on the lookout for that. So we'll see you guys in a very short period of time. Okay, hopefully within the next few days. Okay, have a good evening, afternoon, or morning, depending on where you are. We'll see you guys